Bacterial Taxonomy. At the end of the session, the students will be able to understand the bacterial taxonomy, the classification, and the nomenclature of medically important bacterial infections. First is the classification. So bacteria can be classified based upon the arrangement into taxonomic groups based upon biochemical, physiological, genetic, and morphological properties. So nomenclature means the naming according to the characteristics. Next comes identification. So identification of an unknown taxon and identification of the causative agent of a disease is done. Bacterial classification. So first you should understand the importance of Caliber and Smith's Six Kingdom classification, which was done in 1988, which is the recent taxonomic classification. This one is the most accepted classification and divides all the living structures into six kingdoms like bacteria, protozoa, chromista, plantae, fungi, and animalia. So these are the other classifications. I'll give you a second, just go through. So you can see here that Caliber and Smith in 1998 is the recently used classification. Understood? Student, note down the important points. This will be useful for your future. If you have any queries, you may ask me. Yeah, let's go. Bacterial classification. So the kingdom bacteria is divided into phylum division, class, order, suborder, family, tribe, genus, and species. So this is how the bacterial kingdom is divided into. Okay. So the kingdom will end with the suffix om, class with ia, subclass with id, ae, and so on. You can just go through it. These are self-explanatory, so I don't think I need to explain much on this slide. So the principles which are used for classification of bacteria are mainly three approaches: the phylogenetic, the Adansonian, and the molecular. So this is the hierarchy classification representing the branch which is made into tree-like format. So I have taken so much of effort to make this after reading so much of literature, so make use of it. The phylogenetic classification characteristics are given importance are the morphology, which they can be differentiated into cocci or bacilli. Cocci are round shaped and bacilli are rod shaped based upon the chaining property like gram positive and gram negative according to gram strain. Cultural characteristics like lactose fermenter or non-lactose fermenting colonies on meconic agar. Biochemical reactions, for example COPS and CONS. Okay. Then the antigenic classification like somatic antigen, etc. Your homework is you will find me the full form of COPS and CONS. Understood? And you will let me know in the comment section. Adansonian classification. So this was proposed by Michael Adanson in 1774. This classifies organism giving equal weight to every character. This is used in numeric algorithms like cluster analysis rather than a subjective evaluation. Molecular classification. This is based on the degree of genetic relatedness of different organisms like guanine and cytosine relation with bacteria, and it is estimated after extracting the DNA from a pure bacterial culture. Then comes the nomenclature. So here we are concerned with the designation of specific names of taxa. So this is based on binomial nomenclature which was proposed by Swedish botanist Carolus Linnaeus. I think you have studied about this person in your school period. So binomial nomenclature, here you have the scientific name of a bacteria which comprises of the genus name which starts with a capital letter and a species name. So both the genus and the species are written in italics or they are underlined. For example, you can see Staphylococcus aureus, it starts with Staphylococcus. So इसमें आप देख सकते हो कि स्टैफिलोकोकस में एस कैपिटल है और ऑरियस में ए स्मॉल है, या तो दूसरे टाइप लिख सकते हो विद द अंडरलाइन And genus and species, this was coined based on some properties of bacteria. For example, Staphylococcus aureus was named after its arrangement in clusters. So Staphy means bunch of grapes, and pigments which was found, that was golden yellow pigment, is called aureus. So the bacteria here is called Staphylococcus aureus, which means a golden yellow colored cluster of bunch of grape like appearing bacteria. Fine. So the next example is Neisseria meningitis, which is named after U. Neisser, who discovered it, and the disease which is caused by it is called meningitis. So these are the changes in the name of bacteria. So I will tell you to note down this in your exam purposes, so this might be useful. Okay, so note this down. So these are the name of fungi. So in your exams you may ask, for example, the old name of Penicillium marnefe, which is called as Talleromycetes marnefe. So such questions may be asked, and these two tables play a very important role during your NEET PG examination questions. So kindly note it down. And these are the parasites. So Isospora bellini is now called Cycloisospora belli. Ballontridium coli is now called Neoballontridium coli. Now come the type cultures. So international reference laboratories designated as type culture reference centers. Maintaining the representative culture of established species shows all the standard characteristics of the original strain. The two important type of collections are ATCC, I think everyone knows this, American Type Culture Collection of USA, and NCTC, which is National Collection Type Cultures of the UK. I hope that someone of you might come with some other type of classification and make our country proud. So when whatever you study, get involved in it, do well. Okay. Bacterial infections which are medically important to man. So based upon a gram stain, bacterial infections are grouped into infections caused by gram-positive cocci and bacilli, and gram-negative cocci and bacilli. Okay, this is based upon gram stain, and there are some miscellaneous bacteria which do not take up the gram stain properly. So these are the most important gram-positive cocci which cause human infections. First one is Staphylococcus species. Example is Staphylococcus aureus is most pathogenic organism among the gram-positive cocci. Okay, and next comes the Coagulus negative, which might be a pathogen, or most commonly this is a commensal. Next one is the Streptococcus species, Streptococcus pyogenes, Streptococcus agalactiae, Streptococci enterococci, virulent streptococci, Streptococci pneumoniae. So these are examples. And gram-negative bacteria, see, these are examples. Okay, it doesn't mean that only those organisms in this table cause infection. These are just examples. Just go through. So from the Enterobacteriaceae family, you have see these many bacteria, and non-fermenters like Pseudomonas acinetto, Burkholderia, Stenotrophomonas are examples of non-fermenting gram-negative bacilli which are pathogenic to human. Okay. So anaerobic bacterial infections like spore bearers, examples Clostridium perfringens causing gas gangrene, Clostridium tetani, which is the pathogenic organism causing tetanus, Clostridium botulinum causing botulinum, then Clostridium difficile causing diarrhea in patients who are being chronically administered with antibiotics. Okay, so this causes antibiotic associated diarrhea. So the next comes your spirochetes, rickettsia, chlamydia, mycoplasma, and so on. So try solving these questions. Most recent taxonomic classification of bacteria is based upon, I think all of you know, the answer is Caliver and Smith's Sixth Kingdom classification. Very good, students. Which one of the following is correct sentence? Dr. John will answer. Yes, answer is C. In binomial nomenclature, we have scientific name of bacteria comprising of both genus and species name. Okay, students, thank you for listening. Wish you a happy Sunday. Thank you.